My name's Jamie Padilla, and I am from Albuquerque, New Mexico. I will actually be presenting the evaluation of the single-cell products today. I have no financial disclosures related to this content. This talk is a promotional activity and not approved for continuing education credit. The content of this presentation is my opinions of the product and not of AACR or Illumina. I work for the wonderful Dr. Kim Leslie, and our lab focuses our research on GYN cancers, specifically ovarian and endometrial. Dr. Leslie holds the longest R01 grant in endometrial cancer right now, and she is also the co-lead investigator for the Route 66 Spore Grant, which is currently the only spore grant in endometrial cancer. Our lab is dedicated to bringing more awareness and conducting groundbreaking research in both ovarian and endometrial cancers. The 5-year survival rates have actually worsened from 1970 to 2013 in uterine and cervical cancers, while some of the other cancers like prostate and breast have actually seen positive strides in the 5-year survival of patients. Unfortunately, the mortality rates for uterine endometrial cancer continues to rise to the current decades. So, both endometrial and ovarian cancers are actually among the most underfunded cancers in research, and so this is very important to our lab to change this narrative. Well, one of the major goals in our lab is to actually target mutant P53 and its downstream oncogenic pathways. When P53 is in the wild type state, it's actually the guardian of the genome, and it's a very powerful tumor suppressor. A fun fact about TP53 is that it's actually the most commonly mutated gene in cancer. When it is mutated, it can result in a P53 null state or express a P53 missense mutation, allowing for a gain of oncogenic function. So now that I've talked a little bit about our lab's work and our mission, I'd like to give you a brief overview of my background and its relevance to this presentation. In 2008, I began employment with the University of New Mexico as the senior scientist to the genomics core. At that time, Affymetrix gene chip arrays were all the rave, and it was the main focus of the core. So I actually quickly learned to process that technology. I actually worked on the largest pediatric leukemia cohort for Dr. Cheryl Willman, who was our CEO of the Cancer Center at the time. And I also processed some projects for Dr. Leslie's lab. In early 2011, our core facility added RNA-seq as a service. And it was at that time that I began my journey into RNA-seq and several other next-generation sequencing platforms. In September of 2019, our core acquired the 10x Genomics Chromium machine. It was then that I became familiar with single-cell. I went on to process many single-cell experiments of all different types for the core. I left the core 3 years ago, and I joined Dr. Leslie's lab. And it was a couple months into being with her lab that I was actually introduced to Fluent Bioscience and their team. They presented their product to me, and I was instantly intrigued. So after I was introduced to Fluent and their product, I had some questions for myself. With my extensive background in single cell, the first question that I wanted to answer was, does this technology even work? I mean, will I even be able to produce usable libraries? And my second question was how does this technology compare to 10x? Because I had worked for several years with the 10x product, I knew its capabilities and reproducibilities, but I also knew what its constraints were as well. So I went ahead and did a side-by-side experiment to test these questions. I used an ovarian cell line of ours, and I drugged them with one of our P53 reactivator curcumin analog drugs that our lab studies. And my results ended up answering both of those questions. the Fluent technology worked, and it stood up against 10x. So here I showed a Venn diagram of the genes that were captured on both of those platforms and how many of those genes were actually similar between the two. I pulled out a few of the similar genes because I wanted to look at the log two-fold change between those genes. And as you can see, the numbers were very similar between the two platforms. So I was actually really excited after this, and I wanted to work more with the technology. I knew this technology was going to change things for labs that were unable to actually complete single cell experiments prior to this. So with that, my presentation will highlight the PIP-seq technology, which is now Illumina single cell 3' RNA, and why the emulsion-based microfluidics-free cell approach is going to provide new opportunities to all of those who were unable to compare single cell experiments prior. And like Fiona said, just to let you all know, PIP stands for Particle Templated Instant Partitions. So I'm sure a lot of people are asking, like, why this technology? Well, you know, the Illumina single cell technology, it captured my attention for a few reasons. First, the technology was simple. Any scientist, you don't even have to have any background or prior knowledge in single cell prep, can easily work through this protocol. All you need, and all on your own benchtop. All you need is the vortexer, the benchtop centrifuge, and then just like a small dry bath or thermocycler machine. The protocol has a very easy workflow with some really great stopping points that breaks it up, and it gives you about a 2 to 3 day completion depending on where you want to stop. The next thing is the scalability of the technology. You know, I had several users that came to me when I worked at the core, and they would bring me these really beautiful viable cells, but there was just not anywhere near the amount of cells that we needed to put in for the microfluidics technology. So I'd have to turn them away. I wouldn't be able to process their samples. But with the single cell technology, all of those things are now capable. Our lab, we work heavily with organoids, and so we're able to actually break down those organoids into great single cell suspensions. But again, we just have really small quantities of those cells. So the T2 kit is actually perfect for those experiments. You don't need, you can have just a few hundred cells, and the technology will capture that, giving you great data in the back end. So that's really great. And we also have a collaborator of ours that works with immune cells. And so she was really looking for a much higher cell capture rate to really get that in-depth, deeper analysis that she needed. And we used the T100 kit on some of her samples, and she was able to get some really great data from her ovarian patient samples. And finally, you know, the affordability of the kits that was just the cherry on top for us. You know, with the price of the kits being so affordable, it allowed our lab to actually really scale up the amount of samples that we can now process on single cell. So when Illumina offered me the opportunity to test the new version of the T10 kit, I knew exactly what samples that I wanted to use in the experimental design. My PI, Dr. Leslie, is as part of an IRB approved ovarian and endometrial patient study at the University of New Mexico. The study is actually led by Dr. Sarah Adams, who is a brilliant GYN oncologist. And so the patient samples from this study have been processed and cryopreserved by our human tissue repository over the past couple of years. So I knew that we had samples that were readily available. But for the purposes of the time that I had to complete this, I selected one ovarian and one endometrial patient. to process for this evaluation. So I took a few pictures while I was working through the protocol, just kind of for my own reference. In the first picture, I took a picture of the actual tumor tissue that I was processing for visualization, because I just wanted to compare like the size of the tissue I had and how much of the cellular output I would have on the back end. The second picture is the Illumina approved vortexer with the PIP tubes in it. And really, this is kind of like where the magical moment happens, right? You know, you got your cells in there and you're getting ready to vortex to create those beautiful emulsified single cell capture. And then the last picture is of the samples when you break the emulsion. And for me, that is kind of one of the crucial like moments in the protocol. So I wanted a picture of that. Plus, everyone really likes the pink color that it has. So, and then finally, I'm showing a picture, an image of the QC metrics that was generated from the Illumina connected multiomics software. I worked really closely with the Illumina team in the evaluation of the software, and because I'm not trained at all in bioinformatics, and the software is still being developed, I wanted to ensure that I understood the output of the generated data. So really, my evaluation of the T10 kit was that, and the software was that our samples really performed like quite well. So now I wanted to share a little bit of the preliminary data from these patient samples with you all. This first image here is a PCA plot, and in this plot, both of the samples are represented by two different colors. So the blue dots are representing our endometrial patient, and the red dots will represent our ovarian patient. So I was very excited that we see this like big separation between the two clusters, and because they're two different patients and they're two different types of cancers. So I then wanted to look at each patient sample separately. I was able to create a UMAP of the different graph-based clusters for the ovarian patient sample. And as you can see here, we distinguished 5 different types of cells represented. You know, as I'm continuing to work with the Illumina team, and as they're creating and putting out some of the new features, we'll soon be able to identify each of these clusters. And here's the UMAP for the endometrial patient. So as you can see here, we actually were able to get a few more graph-based clusters represented in this sample. And the expression of the cell clusters are very different from those in the ovarian patient. Like I told you all before, because our lab does study P53, I wanted to see if I could look specifically at the TP53 expression in each of the samples. So here I was able to create a UMAP of the high and low TP53 expressions of the graph-based clusters represented in our ovarian patient. The UMAP was actually, we used it by creating, taking the normalized TP53 counts and splitting the cell population right down the middle. So where you see the low counts are representing the low expression, and the high represents the high expression of TP53. So as you can see here, we have more cells representing the low expression of TP53 than high. And, you know, in the future, we plan to explore this further using the TP53 expression to further classify the cells into populations representing more diversity, and also determine what cell types these clusters are representing. And then here's the UMAP for the endometrial patient with the high and low expression of TP53. So similar to the ovarian patient, our endometrial patient also had more cells that were represented in the low expression of TP53. You know, so as we move forward and we're going to increase the sample quantity for this study, We really hope to dive deeper into the analysis, hopefully finding trends among the different cancer types, and learning much more about the heterogeneity of the tumor microenvironment. So to wrap up my presentation today, I told you about the advantages of the Illumina single-cell technology, which is that it's simple, it's very scalable to help with different experimental designs, and it's extremely affordable. I ended up processing 2 different patient tumor samples to evaluate the T10 kit, and I was successfully able to complete final libraries and very easily. I also successfully produced some preliminary data from these patient samples with the Dragon single-cell RNA and Illumina connected multi-omics software. In the future, our lab will also focus on some different sample types that we will use with these kits, which will include ascites, organoids, PDX, and humanized PDX models. So I'd like to thank Dr. Leslie, Dr. Steinkamp, and Dr. Adams for the support that they gave me to allow me to use these precious patient samples for the evaluation. And of course, thank you to my fellow lab mates for your support, because I know I drove you all crazy during this whole process. So I really do appreciate every single one of you. Thank you, Illumina, as well, and the whole team that worked with me.